Well, greetings, everyone. I'm pleased that you've joined me for Hawthorn University's announcements and interview segment. I'm Paula Bartholomew, and we're just about to begin today's webinar with Dr. Andrea Price. Dr. Price is going to be presenting on nutrigenomics, how food affects our genes. And before we begin our webinars, we usually make time for a few announcements, and we have an interview with Dr. Price. So let me get us started. I'd like to go over just a few logistics because we have a number of new people that are at the webinar for the first time. Participants can ask questions anytime during the presentation by typing them in using your webinar question and chat panel, and we'll save time at the end of the presentation for your questions. And since everyone's muted, I'll present your questions for Dr. Price. We'll also be posting details to the webinar question and chat panel throughout the presentation, and also a few days after the webinar, Alex will email everyone attending this webinar and provide any important details so that you have it and don't have to try to write everything down during it. Also to remind you that this recording is, the presentation is being recorded, and it will be archived and it'll be up on our website under archived webinars in just a few days. And there's a post-webinar survey to fill out after the presentation. That always helps us, and we encourage that. Thank you. And I want to let everybody know that at the last webinar on January 6th, we offered a Hawthorn prize, and we have a winner. So I want to congratulate Tracy Jevning for tuning in to our Food is Medicine webinar with Dr. Price. You were entered into a drawing and you won. So Hawthorn University's 100% cotton tote bag is yours, and it's on the way. Happy about that for you. And I want to congratulate Hawthorn student Tara Sheppy. She's working towards her Master of Science in Holistic and Nutrition Education degree, and she just published the Everything Paleo Pregnancy book. I've got a copy right in my hands that I just received from Tara, and it's available now on Amazon. So if you want to learn more about Tara and the work that she's doing, you can visit her website at whatigather.com. She's got a good blog going, and just a book worth checking out, absolutely. And we have a new feature. We have a Hawthorn webinar discussion forum. It's for Hawthorn students and alumni, and I encourage ongoing discussion of our webinar topics. And so to facilitate this, we've opened up a forum for each webinar. Sometimes there's just questions or comments that we want to continue to engage about, and this is a good platform for us to use. So we always open the post-webinar forum, and all you have to do is log in and type in to participate. And since Dr. Price is presenting this webinar today, we're fortunate that she'll join me to discuss some of her background and interest in nutrigenomics. So hello, Dr. Price. I really appreciate you taking some time and talking with us before you have a presentation to give too.
Thanks, Paula. I really appreciate you giving me this opportunity today.
Absolutely. So let's talk a little bit about nutrigenomics, since that's the topic of the presentation. What initiated your interest? What really sparked your interest in nutrigenomics and epigenetics?
Well, it started when I was a researcher doing the molecular genetics research in breast cancer, and I was just really intrigued by all the different polymorphisms and such that could either increase or decrease risk for disease. They weren't, of course, looking at it from a nutritional perspective, but in terms of just the disease itself. And then I came to learn that, you know, a certain number of those genes could then be affected by nutritional interventions, and so then it just was a natural fit, the two of them together.
Terrific. And have you done any genetic testing for yourself or with your patients?
I have. For myself, I've done the MTHFR testing. I do have a neural tube defect. I was born with spina bifida. My grandmother also has a neural tube defect, and you know, I was born before the 80s. We won't say how far before the 80s, but before the 80s, when they started learning that folic acid protects against neural tube defects. It was in the days of fortification of grains and such, but not with that knowledge. So, you know, I did test myself when I was pregnant for the MTHFR just because of my family history. Fortunately, that wasn't something that I had to contend with because I didn't have the polymorphism for that. I've also done, I do a lot in my practice and on myself with detoxification pathways because I tend to see a lot of environmental medicine clients. And so looking at phase 1 and phase 2 reactions and that sort of thing. And unfortunately for me, I tend to be a slow phase 2 person. So phase 1 is part of the first aspect of liver detox, and it can often turn things into more toxic metabolites. So if your phase 2 is slower than your phase 1, you can end up with a buildup of those phase 1 reactants that are waiting to go into phase 2, but they're wreaking havoc in the meantime. So certain nutrient interventions like EGCG, in green tea, curcumin is a favorite.
And so the first one, AGTC, for people that don't know the acronym, would refer to?
Epigallocatechin gallate. It's one of the main catechins found in green teas. Then curcumin, is a wonderful spice to include. I'm in South Florida where, we have a lot of Caribbean influence, so there's, a lot of curries and things that go on around here. And then, of course, any of your brassica family vegetables that contain those wonderful sulforaphanes will also be helpful in both aspects. They actually kind of slow phase 1 and speed up phase 2, so they're really ideal for someone like me.
Nice, great. Well, I appreciate this information a lot. I think it's very interesting, and I imagine that you're going to go into some more detail about testing and how to apply the information to develop nutritional strategies, either for yourself or for the clients that our students are working with.
Yep.
Excellent. Anything else you want to add about this at this time?
No, I think we're ready to go.
All right. Well, this does conclude the announcements and interview segment, and we'll pause for a few seconds before I formally introduce Dr. Price and we begin her presentation. So give us a minute, and we'll be right back. All right, everybody, it's my pleasure to welcome you to Hawthorn University's Holistic Health and Nutrition webinar series. I'm Paula Bartholomew, and as Director of Online Events, I'll be the moderator for this webinar. I'm here with Dr. Andrea Price, who's our presenter, and will be educating us about nutrigenomics, how food affects our genes. So I want to thank you all for taking the time out and joining us. I want to give you a little bit of background on this topic of the presentation. Over the course of human evolution, diet has profoundly molded human metabolic capacities and thus paved the way for the emergence of modern diseases. There have been numerous studies in humans, animals, and cell cultures that have demonstrated that macronutrients, micronutrients, and naturally occurring bioreactive chemicals regulate gene expression in very diverse ways. And the recognition that nutrients have the ability to interact and modulate molecular mechanisms underlying an organism's physiological functions has prompted a revolution in the field of nutrition. And during this seminar, we'll take a look at how health-gene relationships work and how everybody's unique genetic variations change how our body responds to nutrients in the diet, and how nutrients can affect how our genes are expressed. Our presenter is Dr. Andrea Price, and she's a graduate of Southwest College of Naturopathic Medicine, and following completion of her naturopathic degree, she went into private practice. Dr. Price has a strong background in biomedical research with a significant experience in both benchtop and field research. She maintains a research interest in epigenetics and nutrigenomics, particularly as they relate to oncology. And Dr. Price joined the faculty at Hawthorn University in April of 2014 and also serves on the faculty of Everglades University. And she's recently joined the team at the Integrative Health Institute at Boca Raton, Florida, and is doing a great job there and is a big asset to the clinic. So thank you for that, Dr. Price. It's really a pleasure to have you presenting this webinar, and I know that you've got a lot of information, so I want to turn it over to you and let you begin your presentation.
Great. Thank you so much, Paula.
You're welcome.
Hi, everybody. Welcome to the second webinar of 2015. Today we're looking at nutrigenomics and how food comes to affect our genes. This is kind of a spring off from what we talked about last week with food as medicine. So this is kind of the modernization of that now. Like we talked about last week, since the times of Hippocrates, we've known that nutrition has played a clearly predominant and recognizable role in health management. However, with time and technology, our understanding of diet and its effects on health have evolved from just crude associations to actual conclusive facts, that can be backed up with biochemical evidence. The advent of modern science has led to the realization that not only are certain nutrients essential, but also that specific quantities of each nutrient are necessary for optimal health, and that may differ. Now we know that that may even differ from person to person, even for individuals in the same environment with the same exposures and things. So there's a lot of research going on in this area. It has, the goals, of course, of promoting a personalized, individualized nutrition plans. The dietary recommendations, nutritional epidemiology, and things like that have led us to the newer realization that food directly contributes to disease onset, and that our development of disease as humans can be clearly defined by both environmental influences, our diet, smoking, physical activity, that kind of thing, and hereditary, indicating that it's not necessarily just a nature or just a nurture thing, it's really both together. So nutrigenomics is really one side of a coin with the field of nutrigenetics, and they are slightly different. The nutrigenetics aims to understand how the genetic makeup of an individual coordinates that individual's response to diet, whereas nutrigenomics studies how biochemicals found in foods can alter the genetic expression, the expression of genetic information in each individual. So this leads to the production of different proteins in the body, less, more, not at all, and so that can certainly affect any number of systems in the body, because we have a lot of proteins that are produced. Every enzyme is a protein and things like that. So there's a lot of different ways that a gene being activated or expressed versus inhibited or turned off can change the way our body functions on a molecular level. The aim of both of these fields is to understand the nutrient-gene reactions, particularly depending on the individual genotype, and then of course the ultimate goal being to develop a personalized nutrition strategy for optimal health and disease prevention based on an individual's specific needs. It's increasingly recognized that the nutritional intervention holds great potential for the management of modern chronic disease, and understanding the relationship between metabolism and disease development is a vital foundational tool for nutritional intervention. So these are two really important research aspects that will lead the field of nutrition forward into the future. There's even a school that's developed a Center of Excellence for Nutritional Genomics. It's at UC Davis in California. So, it is becoming increasingly recognized by the Institutes of Higher Learning as well. So this is a graphic that's displaying that, two sides of a coin thing that we talked about. At the top we see nutrigenomics with the food affecting the genes, and then on the bottom we see the nutrigenetics with the genes affecting the way we respond to foods. So, it is a complete cycle that way, and that's why we really have to look at both of them. Our genetics alone do not determine our our future health and well-being. It's really the interaction between genes and environment that do that, and that's including nutrition. And we can see this in certain celebrities, like for example George Burns, a famous comedian, actor, singer, author, who smoked 15 or 20 cigars a day, drank bourbon like it was water, and did this for 70 plus years and lived to be 100 years old. Clearly he had some lifestyle factors that were stacked against him, but his genetics had to be playing a role in him being able to live that type of lifestyle, into a triple digit age. Whereas, most individuals doing those things would have succumbed to any number of chronic diseases long before the age of 100. So to start off, I want to make sure that everybody understands what DNA is and how it encodes our genetic information, and then how we get that information out. So the DNA is a very long polymer that's composed of different nucleotides. The nucleotides are made up of the deoxyribose sugar, a phosphate group, and then one of 4 nucleotide bases. And these bases act like the zipper that kind of hold the backbone of the DNA together. So in the graphic representation that's spinning there on the right, the nucleotides will be represented by the blue and yellow and the red and green circles there. They pair up based on a specific pattern, and they always go together in that specific way. They always pair up as they're set with each other, and each cell has about 2 meters of DNA rolled up inside of it. And the average human has about 75 trillion cells, so that means that's enough DNA to go from the Earth to the Sun more than 400 times. So that's quite a bit of DNA that's balled up inside of our body. We've actually known since the early 1900s that chromosomes carry our genetic information. Chromosomes are made up of tightly bound up DNA, and that's what's known as a hereditary material of humans and almost all other organisms. Every cell in the human body has virtually the same DNA. Most of the DNA is located inside the nucleus of the cell, but there is a small amount that can be found outside that in the mitochondria, which is mitochondrial DNA. Interestingly enough, mitochondrial DNA is only inherited through matrilineal side, so it's only inherited from the mother. So you have the same mitochondrial DNA as your mother, as her mother, as her mother, as her mother. The father has really no input in the mitochondrial DNA. Even more interesting is that all of us humans, our DNA is 99% the same. It's just that very small 1% that makes us different from one another. So back to those DNA base pairs, like I said, they pair up with each other. The A goes with the T and the C goes with the G, and that's how they form the units called the base pairs. And then they attach to the deoxyribose sugar and the phosphate molecule to arrange themselves in the long strands and then spiral to form the helix. So within those DNA strands, that's where our genes are coded. And genes are unique proteins that perform specialized functions in the cell. Our human genome has been identified to contain about 21,000 different genes. So there's a lot of of genes that are in the body. In between the genes are, non-coding base pairs, and there can be errors of duplication, when the DNA replicates in there, that can, affect the way things work. We do have DNA repair enzymes in our body that help to repair that. And then, when these genes are translated into proteins, that's when we get the gene expression. And that produces what's known as a phenotype. And a phenotype is our physical characteristics, what we can see about each other, hair color, eye color, that sort of thing. So the way that the information gets out of the DNA and made into a protein is it first gets transcribed into RNA, and RNA is that single strand there in the middle, the pink one, and that's the process of transcription. This occurs in the nucleus, and the machinery copies the gene sequence into the messenger RNA. It's a molecule that's similar to DNA. It has 4 nucleotide bases, but in RNA, the thymine, the T base, is replaced with a uracil, a U base. So it's slightly different in that sense. After that, the RNA gets translated then into the protein. The protein-making machinery in the cell is called a ribosome. When you used to see those little pictures of the cell in biology class, it was like the kind of round circle with the bumps on it. And that reads the messenger RNA sequence and translates it into the amino acid sequence of the protein. It reads this 3 nucleotides at a time, and these are called a codon. Each 3 nucleotide codon specifies a particular amino acid, and there are stop codons and start codons, so it knows where to stop and where to start, and that helps to tell the ribosome that the protein is then complete. So there can be different levels of interaction between nutrition and genes. These can be direct interactions, so nutrients that interact with their receptor may behave as transcription factors that can then bind to DNA and induce that gene's expression with or without the input of the body. So that can lead to overexpression of certain proteins that we may or may not want. There can also be epigenetic interactions, and these are very interesting. Epigenetics. Epigenetics are changes to the genes that aren't changed in the genetic code necessarily, but these can be passed from generation to generation. So nutrients that can alter the structure of DNA so that gene expression is chronically altered. So that means that if someone was exposed to a chemical and it caused an epigenetic change in their body, they could then pass that change on to their offspring who had never had that exposure. So that's a very interesting field there as well in the epigenetics. And then you can have, genetic variation. So these are common genetic variations like single nucleotide polymorphisms, or SNPs, they're sometimes called. And these can certainly also alter the expression and functionality of our genes. So modifications that can be made by bioactive food components can affect any of those stages of DNA transcription, translation, or later on metabolism once the protein is already formed. It can really occur at any level. Things that can affect transcription would include nutrients like folate, vitamin B12, methionine, choline, and betaine. Those are all nutrients that can affect things like DNA methylation and histone methylation. and that can either turn on genes or turn off genes, and that may affect, the rate at which the those genes are produced. Other water-soluble B vitamins like biotin, niacin, pantothenic acid can also play an important role here. So there's, quite a few nutrients that can affect the turning on or turning off of genes, and food can act at any level of this cascade. So we're going to take a look at some examples of some different ways that food can interact with our genes to either increase or decrease our risk for certain conditions, and how that affects the production of things in our body. So the first one is cancer, and diet can influence our risk for cancer at a number of different stages. Things that are involved with preventing DNA damage, so nutrients like coffee and many of the constituents found in there, curcumin, green tea polyphenols, especially that epigallocatechin gallate that we talked about earlier, abbreviated EGCG, indole 3-carbinol, which is a common nutrient found in your brassica vegetables, broccoli, cauliflower, that kind of thing, a lycopene, pomegranate, resveratrol from grapes, selenium, even soybeans, and vitamin E as well. There's also nutrients that control our regulatory genes, so your omega-3 fatty acids that are in our fatty fish, lycopene from cooked tomatoes, genistein, other soy isoflavones again, and then those cruciferous vegetables really pack an impact on the expression of genes. Hormonal regulation, that can be your soy stuff, your brassica vegetables again, that indole 3-carbinol, DIM, those play a role with the breakdown of estrogens in particular, with the 2:16 hydroxy estrone ratio, and that can either promote cancer or more cancerous metabolite or less cancerous metabolite of estrogen, depending on how the body metabolizes the different estrogens. Ones that are nutrients that are involved with apoptosis, coffee compounds, curcumin, the green tea polyphenols again, selenium, quercetin, which is a nice flavonoid found in things like onions, any of your tocotrienols, which is a category of vitamin E. So those are going to be, in certain nuts, things like that. Vitamin D is also in that category as well. Things that regulate the cell cycle. So apigenin, that's a yellow flavone that's found in many different plants. Aspirin can also be there, although not necessarily a food constituent. The coffee, pterostilbene, which is a very close relative of resveratrol, also found in those grapes and berries and such. Selenium, and then of course any of our cruciferous vegetables too. So there's a lot of bioactive food components that can affect the events that can lead up to cancer. Micronutrient deficiencies are becoming increasingly common in our country. These can be tested for fairly simply for the most part with blood tests and things. You can test for folate levels, you can test for B12 levels. B6 and vitamin C are a little tougher because their water solubility, they're very, kind of in the moment type of nutrients. But, there are tests out there, SpectraCell and such, that test all the different nutrients. And then they can even formulate a custom multiple vitamin or things like that for the individual, if that's what they're interested in. Or from a nutritional standpoint, it does give you sort of a jumping off point for what type of nutrients you would want to focus on for that individual's diet. So folic acid deficiency found in about 10% of the U.S. population. can lead to chromosome breaks, which can increase the risk for colon cancer and brain disorders, neurodegenerative disease, neural tube defects, of course, and that sort of thing. Vitamin B12, about 4% of the U.S. population. They haven't really characterized the type of DNA damage, but it's similar to folic acid, but then you also have the neuronal damage. You get the neuropathies, the numbness and tingling in the hands and feet that come from B12. B6, again, about 10% of the U.S. population. Similar risks to folic acid. People with B6 deficiency might also be more likely to have imbalanced neurotransmitters as well. So you might see those type of issues too. Vitamin C, 15%, incredibly increased risk for cataracts. Vitamin E, about 20%, so a lot of risk for colon cancer and immune dysfunction. Iron, very, very common in women during their menstruating years. Brain and immune dysfunction, cancer is also related there. Zinc, another highly deficient nutrient, and that's the brain and immune dysfunction and also cancers there. So if we focus in on selenium as just one example here, not having enough selenium in the diet can affect any number of genes in the body that are associated with this study with that, as it states on the bottom there, was looking at prostate cancer cells. But it can affect DNA repair, adhesion, and invasion. So that's the ability of a cancer cell to stick on and kind of lay a framework down in another tissue. So that would be metastasis of the cancer. Signal transduction, so that would be sending signals to from the genes to to the protein side of things. It acts as a tumor suppressor, so it can affect those genes that keep our body from getting tumors in the first place. It can affect protein synthesis, growth factors, the cell cycle and apoptosis, angiogenesis, which is the growth of new blood vessels. For cancer cells to grow, they require a tremendous amount of nutrients, and so they also then need a tremendous blood supply to support that. So angiogenesis is a key factor in cancer. And then your transcription factor, so that goes back to the DNA transcription and translation stuff that we talked about earlier. So, one simple nutrient that we can get from dietary sources that can affect so many different aspects of, one of the most frightening diseases that people will come across. We can find it in, whole grains, nuts, seeds, fish, grass-fed and pasture-raised meats. We have selenium from a lot of sources, but it's also a very deficient nutrient. So that's just one example of a mineral that can affect a lot of different genes that are related to cancer. Polyunsaturated fatty acids, these specifically the long chain polyunsaturated fatty acids. These are your EPA and your DHA. And these are what we find in fish oil specifically. We find the shorter chain fatty acids in other nutrients like your flaxseed oil and walnuts and such, which the body does have the ability to assemble, but it's, again, whether it's genetically coded to do that very efficiently where it becomes an issue. There are tests that you can do that can, determine the levels of different fatty acids in the body. And that way you can tell, if someone's on flaxseed oil, you can test and see if they're making enough EPA and DHA from that flaxseed oil. And you can look at all the different breakdown of the fatty acids and know whether or not that's something that needs to be addressed in that individual. So the consumption of the long-chain polyunsaturated fatty acids has led to has been known to beneficially impact a number of physiological processes: growth, neurological development, lean body mass and fat mass, reproduction, immunity, infectious pathologies, the incidence and severity of virtually all chronic and degenerative diseases, things cancer, atherosclerosis, stroke, arthritis, diabetes, any of those types of things. So in the graphic here, you see a giant web, and that's really looking at all of the different ways that the long-chain polyunsaturated fatty acids can affect different genes in the body. And these are genes that even talk to the nucleus in our body, in our cells, excuse me, and they can affect transcription factors, cell regulatory genes, RNA transcription processes. They can affect prostaglandin synthesis. That can have to do with inflammation in the body, inflammation being a backbone for any number of chronic diseases. And then, of course, there are related pro-inflammatory genes as well. So again, that inflammatory factor, which can be related to a lot of issues, from heart disease to cancer and just about everything in between. And as you can see, it's quite a diverse array of genes that it affects. So we've got a number of things there. Just to give you a bit of more information on this graphic here, the green ones are indicative of a downregulation, the red of an upregulation, and the clear of no regulation for a given gene in that system. But you can see that it, does both sides, upregulates and downregulates depending on the gene that it's impacting. So the next thing we have here is the MTHFR. That's the methyl tetrahydrofolate reductase. And this is a gene that is on the chromosome 1 in humans. It's actually subject to an extreme number of polymorphisms, 24 plus polymorphisms. Among the most investigated is the one that we're going to discuss today. That's the C977T SNP. There's a lot of ethnic variability in the frequency of this polymorphism. It's highest in people of Mediterranean and Hispanic descent, next in Caucasian, and then it's lowest in Africans, African Americans, and Afro-Caribbeans. What we have with this is the 677cc, so that's going to be the cytosine, is the wild type. So that's the normal one. That's where people are going to make plenty of this enzyme. You can have the heterogeneous version, which is where they have one T thrown in there. And then these people will function almost the same as normal because they have the C to back up the T that's in there. And this occurs in about 10% of the North American population. And then I noticed a typo right here. We have this one should be 677TT, and that's the homogeneous. This people will have the lowest MTHFR activity. They'll be susceptible to hyperhomocystinemia. Homocysteine is an amino acid that's associated with increased cardiovascular risk, particularly any kind of the vascular type of things, strokes and such, and also increased risk for leukemias and colon cancer as well. So the MTHFR is an example of the single gene polymorphism, and it's an example on the nutrigenetic side. So it's how the genetic code handles exposure to certain nutritional bioactives. So these people wouldn't be able to activate folic acid very well. So supplementing somebody with this polymorphism with folic acid wouldn't be very beneficial because they would not be able to activate it to the methyl tetrahydrofolate, which is what the body actually uses in its cycles. This is something, like I said, that's been very well described in the literature. It is a rate-limiting enzyme in the methyl cycle that converts homocysteine back to methionine, and so it requires methyl donor because it is a remethylation cycle. So it requires a methyl donor, and that's what the 5-methyl tetrahydrofolate is it serves as a methyl donor. Other things that can serve as methyl donors is a methylcobalamin, which is a B12. So again, supplementing these individuals with, if you're using supplemental B12 with, a cyanocobalamin or a hydroxycobalamin would not be ideal because their ability to methylate that would be very low. So genetic variation in this gene can influence susceptibility to, like I mentioned, the occlusive vascular diseases, of course, neural tube defects, Alzheimer's disease, and other forms of dementia, colon cancer, and acute leukemia because of the mutation association there. So a few takeaway messages for this section. Nutrigenomics aims to identify the genes that can influence the risk of diet-related diseases on a genome-wide scale and to understand the mechanisms that underlie these genetic predispositions. Nutrients serve as dietary signals to our genes, and those can be detected by cellular sensor systems that influence what genes are activated and therefore then what proteins are expressed, and then subsequently how our metabolism is able to respond based on those protein productions. The patterns of gene expression, protein expression, and metabolite production in response to particular nutrients or nutritional regimes can be viewed as somewhat of a dietary signature based on that individual. So with regards to testing, there are, as I mentioned, like the MTHFR can be tested by just about any general laboratory. You probably have to, have a physician order those tests. But there is a laboratory called Genova Diagnostics that does a lot of really excellent genetic testing. They have several different genomic profiles. I use them quite often, like I said, for particularly their detoxygenomic profile. And this one evaluates single nucleotide polymorphisms that are associated with increased risk of impaired detoxification capacity, particularly when someone's exposed to environmental toxins. And it can also help to identify people who are particularly susceptible to adverse drug reactions. So, if you have someone that's very sensitive, if I take half an aspirin, I'm knocked out, then you'll, this might be something to suggest for those type of people. It looks at a number of phase 1 and phase 2 genes, and then will help determine if they're, moving faster or slower than what would be expected. They also have a cardiogenomic profile, and this uses genomic testing to identify the risk of genetic susceptibility to cardiac-related diseases. It evaluates single nucleotide polymorphisms that can modulate blood pressure, lipid balance, nutrient metabolism, inflammation, and oxidative stress. So it can give you a lot of avenues with which you could implement nutritional intervention to help balance those things out, or to, keep those genetic overactivities in check, like inflammation and such. There's, anti-inflammatory diets and things that we can implement to help, even if people have a genetic predisposition towards inflammation. They also have testing for estrogen. As I mentioned, that 216 hydroxyestrone ratio really can be one that plays a big role in risk for cancer. So it's something that's very important. Osteoporosis is also in there, as is heart disease to a lesser degree. So the estrogenomic profile evaluates the genes that modulate estrogen metabolism. It also looks into coagulation, because of course that's related to estrogen as well. Cardiovascular function, bone health, and inflammation. So again, a lot of different areas where nutritional intervention can be applied based on the genomic outcome for these people. There are things like 23andMe, and that can kind of give you more of a background in terms of ancestral roots. So you could perhaps guess that certain groups are more prone to this polymorphism or that polymorphism, as we saw with the MTHFR, but it wouldn't give you anything specific for that individual in terms of nutritional type interventions. And then, like I mentioned, there is also that test for fats. That one is something that can look at the breakdown of how the body deals with fats, and how it breaks down and builds up different fats inside the body. It can do the omega 3-6 ratio and that sort of thing. The test is called an omega score test, and it, like I said, can be quite useful for looking at the balance of fatty acids, particularly for people that are only taking those short chain omega-3s. So that's something that you can consider. It can be used to modulate cardiovascular risk as well. I did include several reference sites that have some different articles and such, so if there was anything that piqued your interest in here, the much, the second one on there, this one, and the kaput one down here are particularly interesting ones that I found. And then this Today's Dietitian article is also very, very interesting. So I would encourage you guys to take these references and go check them out for some more information, and to better help you understand this stuff and be able to then apply it in your practices and such as you move forward in your stellar careers that I know you're all going to have. And so that's about all that I have today, and we can open it up for questions whenever we're ready.
All right, thank you, Dr. Price. Yeah, there's a few questions, and I wanted to go back to your the slide that you had on nutrigenetics and cancer. And one of our attendees asked, you talked about cancer and its, talked about coffee and its influence on cancer, and the question is, did you say that coffee helps fight cancer?
Yes.
And so we'd like some clarification on that, of how coffee helps fight cancer.
Sure. Of course, I'm not talking about, your instant Folgers coffee that you grab the powder off the shelf and, mix in, to your hot water. I'm talking about your nice organic coffee that, you would have to brew and things like that. Coffee is a good source of chlorogenic acid, which is, an antioxidant compound and a major polyphenol. So it's actually been looked at in research studies, and you posted in the American Institute for Cancer Research. It was originally thought that coffee might actually increase our risk for cancer, but with further research, it's been shown that the different nutrients in there and some of the other compounds, aside from not caffeine notwithstanding, can actually be of benefit. There are several, it is a tremendous source of antioxidants.
Okay.
Trying to find that. I have a specific article. Yeah, there we go.
There you go.
Here it is. So, yeah, coffee has been shown to also favorably benefit that estrogen ratio with that 2:16 hydroxyestrone ratio. Studies have also shown that coffee consumption slows the growth of estrogen receptor positive breast cancers, particularly those, so that results, then in an older age, a diagnosis, you're not getting so many younger women with cancer. And among women with known cancer-causing mutations, consumption of caffeinated coffee is actually associated with a significant reduction in breast cancer risk. And I'd be more than happy to provide those resources if you'd like. There's somewhere to post them later.
Sure. Yes, we could. You could get those to us, and we can post it with the archived slides of your presentation. Sure. That'd be great. You also, when you were talking about the MTHFR and the specific nutrients of folic acid and B12 supplementing, and certain forms of them wouldn't be beneficial for somebody, but there's a question about what forms would be ideal from these nutrients that would be able to be effectively metabolized if somebody's got a defect.
Sure. So you want to go for the methylated forms. You can buy methylfolate, versus folic acid. And folic acid has been associated with a number of adverse conditions, but that's not the same for the methylated folate. You can just, if you're in supplement form, you can just buy it like that. Of course, folate usually occurs naturally in the foods we eat. It occurs as folate, not as folic acid. So, in terms of dietary-wise, you're already, typically getting the folate side of things. And then the same thing with B12. You want to look for the methylcobalamin versus the hydroxycobalamin or the cyanocobalamin. B12 is also something that I typically, if someone's going to take it as a supplement, that they take it as a sublingual, so as one that dissolves under the tongue, just because it's, somewhat difficult to absorb through the intestines.
Okay. And a follow-up question on the coffee. Is it just caffeinated coffee that has those benefits?
No, that specific study looked at caffeinated coffee, but decaf coffee, may have slightly reduced levels of things like chlorogenic acid, but it would still have some benefits in there. And there's other studies that have found that have highlighted the importance of the non-caffeine components of coffee. So that intake of at least 5 cups per day reduced breast cancer risk by 29%, reduced the risk of difficult to treat estrogen receptor negative tumors by 59%, since most of our therapies for breast cancer are targeted at the hormone positive tumors. If you have those hormone negative tumors, or God forbid the triple negative tumor, then your treatment options are tremendously reduced. And the coffee, the 5 cups per day, also reduced the risk of postmenopausal breast cancer by 37%, excuse me.
I'm sorry, but it probably reduces their ability to sleep too. like that is a lot of coffee, and uh, I, I don't know. I've looked at a number of studies also that caution against coffee and adverse effects. So it comes back to the study and what the parameters are, and so many factors there.
Yep.
So you also you were talking about the percentage of the U.S. population that had nutrient deficiencies, and there were a number of us that were really surprised that the deficiency. Thanks for pulling up that slide. That were surprised that the deficiencies weren't higher, and I'm um, wondering
Well,
what the.
they probably are.
Mhm,
These are people that participated in a study. So, you know, there's, they may have been, picking people from, they might not have been picking people that are from, certain socioeconomic statuses that would be, more prone to micronutrient deficiencies,
mhm,
because of their low intakes of the fruits and vegetables.
mhm,
Um, and things like that. They might have, picked everybody from the Upper West Side of Manhattan, to do the study where, the people would tend to eat more whole foods diet, organic foods, and things like that. It might not be, um, and they didn't share in the study the source of the participants. They just, give the number and such, but not where they source their participants from. I agree. I would expect them to be much higher.
Yeah, and so I'm glad we asked because it's really an important qualification around it that this is a
And
specific population as part of a study, not necessarily a reflection of
not
U.S.
generalizable to
population.
the, yeah.
Yeah,
And bear in mind, this is also talking about RDA, which is pitifully low anyways. I mean, I think the RDA for vitamin C is like
six.
26 milligrams or
Yeah,
something.
yes.
It's ridiculous. It's like 2 milligrams more than you need to keep from getting scurvy.
Well, I think that's what the baseline is just the amount that you need to prevent disease or
Yeah,
symptoms of disease coming up. So there's those markers rather than
than looking at it from a health
optimal
standpoint.
reference
Yeah.
intakes. Absolutely, yes. And you also talked about with your B6 and folic acid, do you have, with the methylated form, do you have dosages that are commonly recommended for that?
Well, with the methylated forms for a woman that is postmenopausal or or is um, or men, you would need about 400 micrograms per day.
Hmm.
And for a woman that's in her childbearing years who, there's a chance that she could become pregnant or is trying to become pregnant, you would want to err on the side of caution and go with 800 micrograms per day minimum.
Hmm.
I mean, when I was in the peri-conception time for my son, I took upwards of 5 milligrams because I was really paranoid about the MTHFR because that stuff happens in the very, very early stages, in the first, 3 weeks or so of pregnancy. So that's when you need the folic acid the most. So you really need it, kind of before you're even pregnant. And they've done several studies that show that the folic acid status of the mother around the time of conception can lead to decreased cancer risk and decreased chronic disease in the offspring.
Hmm. All right. Um, there's a question here about Alzheimer's disease, and I think it's an important one here when we talk about theories because certainly genetics is one of the common theories that plays into the development of Alzheimer's, and research in the 90s linked a gene called the apolipine E gene, short version we call ApoE gene. And there's three variations of this gene. There's E2, which is noted to be the most protective form from Alzheimer's, and
Hmm.
there's E4, and when E4 is present, it's much more likely that Alzheimer's will generate. And then there's E3, it's the most common of the gene, and it's linked to possible risk of Alzheimer's. So you come back to the beginning of your presentation where we inherit two copies of our genes, one from each parent. So there's 6 possible combinations of these genes, and the worst combination would be if someone would have 2 of the E4 genes.
Sure.
And yeah, actually, I think up to 3% of the population are known to have these genes, and they're 90% likely to have Alzheimer's by the time they reach 80 years old. And so the best and the most protective combination someone could have would do you have a set of E2 genes.
Yeah.
That's uncommon, but it's the
sure
most
there's somebody.
protective from it. Yeah, I hope so. So yeah, and then as you've discussed, there's theories that that because the brain can be exposed to a variety of negative forces and environmental factors, that some of these actually have the ability to kill neurons directly, and also to create free radicals and observe normal brain cell function. So it's with Alzheimer's, just such a huge epidemic in our culture, and earlier onset of dementia being seen. I think it's an important factor to bring up.
Absolutely. Humans are exposed at this point in time to more chemicals than we've ever been exposed to in our history. And the powers that be and the EPA and such, oh, it's just a little bit. Oh, it's just a little bit. Well, it's just a little bit of thousands of different chemicals, and the additive effects of these things is really unknown. We have no idea what that does to the body. These epidemic rates of, neurological diseases, with Alzheimer's and, autism is now skyrocketing. And I don't think all of that can't be due to better diagnostic methods. When I was in school, I never saw children, or maybe one, that had so was on the autism spectrum. Whereas now that there's so many, and it's kind of scary.
Yes, yeah, it's quite alarming, and it's good that we do the work that we do, because there's big work and help that's needed.
Absolutely.
Can you talk a little bit about any specific superfoods that might help to protect us from overall gene mutations?
Sure. Well, of course, my personal favorite is going to be the broccoli, or, any of those cruciferous vegetables. I just like broccoli better than cauliflower. So yeah, any of those, are going to be nutrients that are going to contain the nutrients that can help protect our DNA in a number of ways. They help to prevent DNA damage. They control those regulatory genes. They fight cancer promoting inflammation. They can block excessive cell replication, which is, of course, the hallmark feature of cancer. And they can, trigger the death of cancer cells, through the process of apoptosis. And they can also help restore receptors to enhance standard treatments. So, if someone did have, like, for example, the breast cancer, and estrogen negative cancer, then these, the cruciferous vegetables can help to modify and restore breast cancer hormone receptors, which can then increase their susceptibility to our current therapies.
Thank you. There's a couple more questions if you have time. Right. The surprise that we haven't heard more about the relationship between true vitamin A and vitamin D and gene expression. Do you think there's something that needs to be discussed further there?
Well, vitamin D certainly, plays is known to affect hundreds of genes in the body. So, and it's wildly deficient. Like I said, I live in South Florida, where we our quote unquote winter is when it gets down to 60 degrees here.
For you.
So we always have sun. There's plenty of sun all the time. Yet DNA, our vitamin D is very low. I test it routinely in my patients at the clinic, and I've yet to see somebody who's not supplemented who has optimal levels of vitamin D.
Yeah.
I'd like to see it between 50 and 80, and usually I get 30. I mean, like a high one is if I get a 42. And these are people down here are very outdoor. Half your lifestyle is outside because the weather is so nice for that sort of thing.
Mm-hmm.
And I still see that very low vitamin D. Got this reputation as a a bone vitamin, but nearly every tissue and cell type in the body actually has receptors for vitamin D. So I think that discovery has really radically changed how we've come to be looking at the role of vitamin D in the body. Having optimal levels has been shown to benefit a host of disorders from heart disease and Alzheimer's to diabetes. So there's really a lot of ways that that vitamin D can help the body.
Okay. There's a question that's specific to MTHFR and supplementation, and with specific questions that come to the individual, it's hard sometimes to get specific, and I just put that in perspective when I ask. This person has the MTHFR gene mutation C677T heterozygous and A1298C heterozygous. How bad is this on the scale, and is it sufficient to take methylfolate 1,000 micrograms and methylcobalamin 1,000 micrograms? Doesn't seem to be a clear answer as to how much is necessary, and be taking a certain type of B6 to improve their effectiveness.
Sure. Yeah, there's not a real concrete answer as to how much one would have to take. I typically suggest about 5 milligrams total. So, they're at about 2 with that protocol. But given that's for the homogeneous, so given that they're the heterozygous, their function for the 677 is nearly normal because of having that C in there. Will certainly help them in that aspect. So I would say about what they're doing is probably sufficient, particularly if they're coupling it with the whole foods type of diet.
Okay.
Fruits and vegetables and things like that.
And is there B6 to improve effectiveness? Is that helpful?
The pyridoxal 5-phosphate, P5P, is pretty much the best one that we can get these days, since they've taken pyridoxine off the market. They've taken the other forms off the market to develop them into drugs.
Thank you for that. I'm so grateful for all that support. And last question here, going back to vegetables. And the cruciferous type. Are lacto-fermented vegetables more suitable in this situation than fresh ones? Would they confer any more health benefit or protection?
Well, I mean, I guess, because they would have the amino acids, and the cell wall would probably be, either weakened / broken, due to the fermentation process and the, somewhat of acidity that's created during that. So perhaps, yeah, more nutrients would be available. I haven't looked at any studies specifically, but just considering just the mechanical standpoint, breaking down the cell walls of plants and being able to, extract the nutrients from inside of there, is really key when we're talking about gaining nutrients from plant sources. And, our teeth can do a fairly good job of it, but, we're talking cells. So, those are very, very tiny. So, yeah, I would think that using fermented foods would make it easier to access the nutrients because the cell walls would be either broken or weakened, and then you also have those added probiotics in there as well.
All right, thank you. Do you have any closing comments, Dr. Price, before we go to closing comments?
No, I'll just provide those additional coffee references for everybody to take a look at.
Okay.
you want me to just add another, can I just add another slide onto this with the coffee
Sure.
ones, or?
Absolutely, yeah, that would be
Okay,
fine. Yeah,
so I'll
that's
just do
easier.
that and then I'll send you the final.
Okay. All right, great. All right, let me take care of some closing comments. I want to remind everybody that this webinar was recorded and it'll be available on the Hawthorn website and under archived webinars in just a few days. And so you'll be able to review this and a lot of other topics that have been posted there as well. And if you're interested in Hawthorn's program and then the courses that we offer, there's a lot more detail on our website. And Kathy McDermott's our Director of Admissions and always happy to answer your questions. There is a survey to fill out after the webinar ends. It really helps us to have your feedback and your comments. We do take it into consideration carefully, so we appreciate you taking the time. I want to let you know about our next webinar and invite you to join us on Tuesday, February 3rd. We are going to host adjunct faculty member Glenn Depke, and Glenn's going to be presenting on adrenal fatigue. It's a rising epidemic, and we'll start at 3:50 for our announcements and interview segment with Glenn Depke, so I hope you'll join us. And this does conclude today's presentation. I want to thank you again, Dr. Price. It was really a pleasure to have you with us 2 times
Thanks
in a
so much,
row.
Paula. Have a great day.
All right, you too. And I want to
Thanks.
thank James and Alex for all your help and to everyone for sharing this educational experience with us. I do want to wish you all the best of health and hope you'll join us for Hawthorn University's next Holistic Health and Nutrition webinars. And until then, be well and take good care of yourselves.